Hey guys, in this video tutorial we'll be learning about CFTR. So CFTR is abbreviated as cystic fibrosis transmembrane conductance regulator. Basically it is a protein which acts as a chloride transporter. So this CFTR present in different organ of the body like lining surface of the alimentary canal, pancreas, lung, and it acts as a chloride ion channel. Here we will see how it works in the enterocytes. So basically enterocytes are the absorptive cell, that is simple columnar epithelial cell which lining the surface of small intestine. Here the CFTR help in osmosis of the lining of the small intestine. So let's see the mechanism behind it. In the enterocyte, the chloride ions are influx into the enterocyte from the outside by the exchange of sodium and potassium ion. There is a ligand known as VIP. VIP is known as vasoactive intestinal peptide. So when this VIP bound to the VIP receptor, this is a GPCR receptor. The VIP receptor is also known as a GPCR receptor. So when the VIP bound to the VIP receptor, the GS subunit active from the trimeric alpha beta gamma subunit. So this GS again activate the adenylate cyclase. Then the adenylate cyclase convert the AMP to CAMP, adenine monophosphate to cyclic adenine monophosphate. Then this CAMP activate PK in the downstream protein kinase A, and the role of the protein kinase A is to phosphorylate the CFTR transporter. This is the CFTR transporter. The phosphorylated CFTR transporter, the phosphorylated CFTR can transport the chloride ion from the enterocyte to the lumen of the intestine. This chloride ion has a property, that is it is act as a osmotic agent. That is when the chloride is transported to the luminal site, it help in the transport of mucus and water to the luminal site, so that the total lining will be hydrated, and in the hydrated environment this microvilli can move properly. So there is different diseases and disorders linked to the CFTR when there is mutation in the CFTR. Like when the CFTR channel is mutated, there is dehydration in the luminal site. Besides this, the dysregulation of epithelial fluid transport due to the mutation, and in the vast difference, if there is mutation in the CFTR, then it will lead to male infertility due to congenital absence of vast difference. Like also it cause the disease like diabetes, malnutrition, then respiratory infections, and it also cause less life expectancy. So I would like to request you to note the information and try to solve this question. So in this question it's given that the cystic fibrosis transmembrane conductance regulator is known to control the transport of which ion. This is a straightforward question and the answer is D, the chloride ion. So it's a question from CSIR net June 2012. It's given that the CFTR protein is known to be a CAMP dependent chloride channel in a patient with a mutant CFTR shows chloride permeability reduced, and as a result it exhibit chloride level in the sweat. Okay, the to prove this CFTR protein, both wild type and mutant are inserted in a model membrane liposome. So inside the liposome they have given the mutant and wild type, and the chloride transport followed with radioactive chloride. The chloride ion is radioactive. It is known that the topology of CFTR in the membrane is very important for its function. Despite no proteolytic degradation or denaturation, the CFTR protein in the wild type CFTR failed to transport Cl in the liposome. So if in the wild type CFTR it is failed to transport the chloride in liposome, then there can be two possibilities. Either there is a problem in the GPCR pathway which failed to phosphorylate the CFTR, or the insertion of the CFTR protein in the membrane is in the false position or wrongly inserted. So it's asking which of the following is the correct explanation for this, and it's clearly seen that C, where it's given the option is the CFTR protein gets wrongly inserted in the liposome. Thanks for watching the channel. If it is in any way helpful to you, then please do subscribe the channel, and for more updates like this please hit the bell icon.